The overall goal of this procedure is to use a gliding assay to measure the flexural rigidity of microtubules. This is accomplished by first specifically attaching the motor protein kinesin to a microscope slide with motor domains free to bind microtubules. The second step is to add fluorescently labeled microtubules to the sample, which are translocated by the kinesins. Next, videos of gliding microtubules are collected using fluorescence microscopy. The final step is analyzing the gliding assay videos to calculate the microtubule flexural rigidity. Ultimately, the gliding assays are used to determine the flexural rigidity of single microtubules.
This method can help answer key questions in the field of cytoskeletal mechanics, such as the bending stiffness of individual cytoskeletal polymers. Demonstrating this procedure will be Anna Ratliff, a student from my laboratory.
To begin this procedure, prepare the working assay buffer by adding 10 microliters of 200 mmol dithiothreatol, or DTT, to 1 milliliter of the stock assay buffer solution and mix. Keep this and all other working solutions at room temperature during the procedure. Next, using the working assay buffer, prepare 40 microliters of biotin BSA buffer by combining sterile filtered biotinylated bovine serum albumin at 2 mg per milliliter with the working assay buffer in a 1:1 ratio and mixing the solution. Then, prepare the BSA buffer by combining 645 microliters of assay buffer with 5.5 microliters of 117 mg per milliliter BSA and mixing the solution. Also, prepare the streptavidin buffer by mixing 57 microliters of assay buffer with 3 microliters of sterile filtered streptavidin at 10 mg per milliliter in assay buffer.